In this question, you have to match the items given in column 1 with those in column 2. Now, citric acid is industrially produced by Aspergillus niger. Thus, A matches with 3. Now, cyclosporin A is an immunosuppressant drug that is produced by Trichoderma polysporum. Thus, B matches with 1. Now, statins, which is used to lower the cholesterol, are derived from Monascus. Thus, C matches with 4. And butyric acid is produced by Clostridium butyricum. Thus, D matches with 2. Hence, the correct answer is option D. Biochemical oxygen demand, that is BOD, may not be a good index for pollution for water bodies receiving effluents from option A, petroleum industry. Now, biochemical oxygen demand measures the amount of oxygen required by microorganisms to decompose organic matter in water. And effluents from the petroleum industry contain non-biodegradable hydrocarbons and other chemicals which do not significantly contribute to BOD. So, making it unsuitable index for pollution from this source. Hence, it is the correct option. Option B, sugar industry. Now, sugar industry effluents are rich in organic matter, which increases BOD due to microbial deposition. Hence, it is incorrect. Option C, domestic sewage. It is also incorrect because domestic sewage contains high amounts of biodegradable organic matter, making BOD a reliable indicator of pollution. Option D, dairy industry. Now, dairy industry effluents also contain biodegradable organic substances like proteins, fats, contributing to high BOD levels. Hence, it is also incorrect. In this question, you have to identify the mismatch pair. Option A, Rhodospirillum is to Mycorrhiza. Now, this is a mismatch because Rhodospirillum is a genus of the photosynthetic nitrogen fixing bacterium and it is not associated with mycorrhiza. Mycorrhiza refers to a symbiotic association between fungi and plant roots. Hence, it is the correct option. Option B, Anabina is to nitrogen fixer. Now, this is a correct match because Anabina, which is a cyanobacterium, it is known for its ability to fix nitrogen. Hence, it is incorrect. Option C, Rhizobium is to alfalfa. Now, this is also a correct match because ribosome forms a symbiotic relationship with legumes, including alfalfa, to fix atmospheric nitrogen. Hence, it is incorrect. Option D, Frankia is to Alnus. This is also a correct match because Frankia, which is an actinobacterium, it forms symbiotic nitrogen fixing nodules with non-leguminous plants like Alnus. Hence, it is also incorrect. In this question, you have to identify the step in sewage treatment that removes suspended solids. Option A, secondary treatment. It is incorrect because secondary treatment, it involves the biological breakdown of organic matter using microorganisms to degrade dissolved organic substances. Option B, primary treatment. Now, primary treatment is the first stage of sewage treatment, which involves the removal of suspended solids and organic matter through physical processes such as screening, sedimentation, and flotation. Hence, it is the correct option. Option C, sludge treatment. It is incorrect because Sludge treatment occurs after the primary and secondary treatment stages. It involves the processing of the solid waste, that is the sludge, that is generated during the earlier stages. Option T, tertiary treatment. It is also incorrect because it is the final stage of sewage treatment, and it is for the further improving of water quality by removing the remaining contaminants such as nitrogen, phosphorus, or remaining pathogens. In this question, you have to identify the correctly matched pair. Option A, methanobacterium is to lactic acid. It is incorrect because methanobacterium produces methane gas and not lactic acid. Lactic acid is produced by lactobacillus. Option B, Penicillium notatum is to acetic acid. It is also incorrect because Penicillium notatum produces antibiotic penicillin and acetic acid is produced by bacteria like Acetobacter aceti. Option C, Saccharomyces cerevisiae is to ethanol. Now, Saccharomyces cerevisiae, also known as brewer's yeast, is used in fermentation processes and it produces ethanol and carbon dioxide. Hence, it is the correct option. Option D, Acetobacter aceti is to antibiotics. It is also incorrect. As already stated, Acetobacter aceti is involved in the production of acetic acid and not antibiotics. In this question, you have to point out why the conversion of milk to curd improves its nutritional value. Option A, by increasing the amount of vitamin D. It is incorrect because vitamin D levels generally remains unaffected by the process or in the process of conversion of milk to curd, that is fermentation. Option B, vitamin A. It is also incorrect. Vitamin A, which is also a fat soluble vitamin, is not significantly changed by the fermentation process from milk to curd. Option C, vitamin E. This is also incorrect because like vitamin A or vitamin D, vitamin E is a fat soluble vitamin and is generally not affected by the bacterial fermentation process which converts milk to curd. Option D, vitamin B. Now the conversion of milk to curd or yogurt involves the fermentation by lactic acid bacteria, mainly lactobacillus species. So vitamin B12 or vitamin B2, that is riboflavin, are produced in higher quantities or amounts during the fermentation process. Now this enhances the nutritional value of curd as compared to milk. Thus, it is the correct option. In this question, you have to choose the option that includes the correct group of biocontrol agents. Option A, Bacillus thuringiensis, tobacco mosaic virus, and aphids. While Bacillus thuringiensis is a biocontrol agent, but tobacco mosaic virus is a plant pathogen and not a biocontrol agent. Aphids are pests and also not biocontrol agent. Hence, it is incorrect. Option B, Trichoderma, baculovirus, and Bacillus thuringiensis. Now, Trichoderma is a genus of fungi used to combat soil-borne plant pathogens, and it works by parasitizing harmful fungi and stimulating plant immunity. Thus, it is a biocontrol agent. Baculovirus is a type of virus used to target specific insect pests, and they are host specific and as well as environmentally safe and are often used against caterpillar pests. Thus, it is also a biocontrol agent. Bacillus thuringiensis is a bacterium that produce a toxin known as Bt toxin, which is harmful to many insect larvae, and it is commonly used to protect crops from pests like caterpillar. Thus, it is also a biocontrol agent. Hence, this group contains all the agents which are used for biocontrolling in agriculture to control pests and pathogens. Thus, it is the correct option. Option C, Ocetelluria, Rhizobium and Trichoderma. It is also incorrect because Oscillatoria and Rhizobium are involved in nitrogen fixation and not as in pest control. And Trichoderma is a biocontrol agent. Option D, Nostoc, Azospirillum and Nucleopolyhedrovirus. It is also incorrect because Nostoc and Azospirillum are nitrogen fixing bacteria and not biocontrol agent. But this virus can be used for pest control. In this question, you have to match the components given in list 1 with that of list 2. Lactobacillus species are commonly used in the fermentation of milk to produce curd. Thus, A matches with 2. Saccharomyces cerevisiae, which is commonly known as baker's yeast, is used in the fermentation of dough during bread making. Thus, B matches with 4. Aspergillus niger is one of the primary organisms which is used industrially to produce citric acid through fermentation. Thus, C matches with 3. Now, Acetobacter aceti is widely used in the production of vinegar. Thus, D matches with 5. Hence, the correct answer is option B. In this question, you have to identify the equipment that is required for growing microbes on a large scale for industrial production of enzymes. Option A, BOD incubator. Now, a BOD, that is biological oxygen demand incubator, is used for testing the oxygen demand of bacteria in water samples, and it can be used for environmental monitoring and research purposes, but not for the industrial production of enzymes. Hence, it is incorrect. Option B, sludge digester. It is also incorrect because a sludge digester is used in wastewater treatment processes, but it is not used for growing microbes for enzyme production. Option C, industrial oven. This is also incorrect because an industrial oven is used for purposes like drying, baking or curing materials, but it is not suitable for cultivating microbes. Option D, bioreactor. Now, a bioreactor is a specially designed vessel that provides the necessary conditions such as optimum temperature, pH, and proper aeration for the growth and multiplication of microbes on a large scale. And so, this equipment is extensively used in the industrial production of enzymes, then pharmaceuticals and other biotechnological products. Thus, it is the correct option. In this question, you have to identify the compound which is used as a commercial blood cholesterol lowering agent. Option A, cyclosporin A. It is incorrect because cyclosporin A is an immunosuppressive drug which is commonly used to prevent organ transplant rejection, but it is not used to lower the blood cholesterol levels. Option B, statin. Now, statins are a class of drugs which are used to lower cholesterol levels in the blood, and they work by inhibiting the enzyme HMG CoA reductase, which is involved in cholesterol synthesis in the liver. So, this reduces the overall cholesterol levels and helps to prevent cardiovascular diseases. Thus, it is the correct option. Option C, streptokinase. It is also incorrect because streptokinase is an enzyme which is used to dissolve blood clots in cases of heart attack or pulmonary embolism. But it is not related to lowering the cholesterol levels in blood. Option D, lipases. It is also incorrect because lipases are enzymes that break down fats into fatty acid and glycerol, and they are involved in digestion but are not used to lower cholesterol level in the blood.